Well, good morning, small group. This morning I'm Sam Rushforth, Dean of the College of Science and Health, and this is part of our two-day symposium from the different departments, kind of a showcase. You're in for a treat this morning from Dr. Tulane, and so Dr. Heaton is going to introduce him. But we have excellent speakers all day today. This is going to be a fine day. And Dr. Arden Pope is coming at noon. He's one of the very foremost air pollution researchers in the world, and he's going to talk about the impact of air pollution on our health. And we have plenty of air pollution, of course, in this valley. So welcome to this particular event, and I'm going to tell Darren to come up, but please come all day if you can and enjoy yourself. Thank you.
Okay, you guys are fortunate you get a small group, so it should be a really interactive discussion. I'm sure Dr. Tulane would welcome any questions that you have as he does his presentation. Just let me say a little bit about Dr. Tulane. He's such an excellent teacher, and most of you are probably students in his class, so you know that. But we sure have appreciated him in the department. He's an excellent teacher and is probably the best researcher in our department also. We're glad to have him, we're lucky to have him, and I'll turn the time over to him.
Well, thank you, Darren, for those kind words. I'll make sure that I remind you of them. I hope that they were videotaped when it comes time for any difficulty, with the U.S. chair and my need to convince you that I ought to be kept, even though you might think otherwise at some point. But so I am representing the chemistry department, and so on the title slide I thought I'd put a picture of me looking like a chemist. Which those are more rare pictures than most of the pictures of me. But I also decided to include whatever the heck this is from the advertisement for the seminar. I think that's supposed to be DNA, but it doesn't actually work as DNA. It's going the wrong direction and a bunch of other things. But anyway, I just wanted to make sure that you all knew that I don't know what the heck that is. So today I'm going to be talking about top-down biochemistry and its use in looking for disease biomarkers. And I'm going to start by talking about what it means to be top-down and how that's compared to, contrasted to the way a lot of science is done. Now, these terms, hypothesis-driven or bottom-up and discovery-based or top-down, don't apply just to biochemistry or even to biology or chemistry. They're really broader than that, but they've become more talked about and more obvious to people that practice biochemistry and biology lately. Hypothesis-driven, bottom-up science is what you've probably heard the most about, but discovery-based or top-down science is becoming more and more important. So, wrong button. I'll get this down by the end. So hypothesis-driven science is what you've probably heard described as the method that science has done since you were pretty young, Hypothesis-driven science is the forming and testing of hypotheses, and it's what we sometimes refer to as the scientific method, as if there's only really one narrow way to do science. Since science is, in the words of Albert Einstein, just a refinement of everyday thinking, and basically science is anything that you can do where you can actually empirically test your statements about the world. It's broader than the scientific method as you were taught in elementary school, but the method that you were taught in elementary school as the scientific method includes, as a very important element of it, hypothesis-driven science. And this is also called bottom-up science because you tend to work from more details up to a larger picture and make theoretical constructs that are hypotheses and can become theories if they're supported. if you find patterns and abstract conceptual explanations for the data and the observations that you collect. And this is to be contrasted with what we now refer to as discovery-based science or top-down science. Basically, discovery-based science is, as that name implies, just going out into nature and discovering what's there. Go out and find out what nature actually contains. And this has been done for a long time, even though the use of the term is really more recent in terms of how often we refer to it. But it was done, for example, for the last 100 years or so as genetic screening. And this is a way to find out what biological functions are encoded by genes, and to identify the genes that encode different biological functions and enable us to study how a biological function is accomplished by an organism. Biological and geological surveys are another example of discovery-based science that have gone on for a long time and are very important in terms of enriching our understanding of nature. These discovery-based methods, though, were often referred to pejoratively as fishing expeditions, and in fact they were really difficult kinds of things to get funding for. It was difficult to convince the NIH or the NSF that it was worthwhile to spend money just to go out and find things when you appeared not to know what the heck you were looking for. But this has changed a little bit because we've realized that instead of imposing a bias on nature, top-down approaches can actually really help us to learn in ways that we couldn't learn if we're trying to pretend that we know the answers before we go looking for them. Darren?
Sure, not the first person that's had this thought, but in essence, Darwin's observations were discovery-based.
So systematics and the biological surveys that lead to understanding what is out there are a really important part of understanding evolutionary biology. But like almost all fields of not only biology but science broader, there is a conversation between hypothesis-driven and discovery-based approaches. And so it took naturalists looking into nature and seeing what there was, but also theorists, which Darwin was a great example of, both a naturalist and a theorist, making hypotheses to try to account for what was observed. So he did some of both, and evolutionary biology is one of those fields that's very well founded on a mixture of the two approaches, and I think that you'll agree that both have something to offer. Discovery-based approaches enrich our ability to make hypotheses that we can do bottom-up biology with, or other types of science. Okay? So I mentioned how discovery-based science was difficult to get funded, but that changed about the time that I was in the shoes of some of you folks that are getting your undergraduate degree, as the newest kid on the block in terms of significant public funding of science came along, and that was the Human Genome Project, which started in the late 1980s. The Human Genome Project became the poster child for discovery-based success. So in the year 2001, early 2001, the first publications of the human genome at a preliminary level were made. Both the public genome effort that was published in Nature magazine and the private effort that was published in Science magazine, both of them led by these two gentlemen on Time magazine here. Craig Venter leading the private effort at Celera Genomics, and Francis Collins at the NIH leading the public effort in cooperation with a lot of other people. And here the two of them are with some guy, Bill something or other, who presented them as wanting to fill the shoes of William Jefferson, his namesake, I guess, not William Jefferson. His middle name was Jefferson, so I got confused. Thomas Jefferson, with Collins and Venter in the roles of Lewis and Clark here, saying that this was as important a map, if not more important, than the maps that were produced by their expedition. And the expedition that was known as the expedition, the Lewis and Clark foray, was a good example of discovery-based science as well. So the success of the Human Genome Project, the fact that we learned so much, among the things that we learned, the most important thing that we learned from the Genome Project was that we understood so little about what we thought we understood. That was a very revelatory and very important step forward, and it opened the eyes of the government funding agencies to the value of discovery-based science. And so discovery-based science has been a little bit easier to fund since then. And after the genome was finished and people were really high on the idea of discovery-based science, then the question was, well, where do we go with this enthusiasm and with the newfound information that we have because of the genome? And one of the places that was a significant new direction enabled by the success of the Genome Project was proteomics. So proteomics is the study of the totality or complement of proteins in a cell or organism, just like genomics is the study of the genetic materials, entire complement in a cell or organism. And it's been defined a number of different ways, but my favorite way that proteomics was ever defined was defined by Dan Charles on a NPR radio report. Now that you know that I listen to NPR and that you can see that I have a beard, you know that I'm a liberal. While I was driving down the road one day, I was listening in 2002, listening to the radio, and I just about had an accident when all of a sudden there was a radio report about the field of biochemistry in which I work, proteomics. That was pretty weird. It was almost surreal. I for a minute wondered who had hacked into my radio. But Mr. Charles was reporting on some of the early and important proteomics work that was being done to understand how different networks of proteins works, and to understand what proteomics itself was, to help his audience understand. Dan Charles referred to proteomics as going to parties, where what the genome provides us is a guest list of all of the different proteins that can be invited to parties, and proteomics is to actually go to the parties and find out who's there, what they eat, who they sit with, and whether or not they have a good time. And since I was excluded from a lot of parties earlier in my life, I decided that I really liked this analogy, that I, for a living, crash parties. That really sounded like a good idea. Never mind that these parties are really small and kind of conversationally boring and everything. They're parties nonetheless, and I get to crash parties for a living. So I like that idea. Another example of how important proteomics was becoming was the year before, in 2001, the Human Proteome Organization, or HUPO, was started by having this initiating conference, the Human Proteome Project Conference. And I love the subtitle of their conference, Genes Were Easy. Genomics is much easier than proteomics, but the idea is that proteomics, because you're actually going into the realm of the function of the gene products, then you're actually going to learn something more meaningful and be able to advance our understanding even more than genomics could. So genomics and proteomics started a list of omics, and for a time the suffix omics seemed to mean fundable. And so if you could append that suffix to whatever you were doing, then you could make it easier to, and this was exactly the opposite of the fishing expedition perspective on discovery science that had prevailed up until the Genomics Project. So genomics is the study of the full complement of genetic material. And proteomics is the study of the full complement of the proteins encoded by that genetic material in any given circumstance or given biological tissue. Glycomics is study of the carbohydrates, especially the carbohydrates attached to proteins, because it's the proteins that are really important, of course. As a protein chemist, I have to say that. Lipidomics, the study of lipids, and I'll refer to this a little bit. Transcriptomics, the study of the RNA transcripts that are made from the genome, also a vibrant and active field. And then a field who doesn't really know what its name is, metabonomics or metabolomics. I've heard people that do this call it by both. And I tend to favor metabolomics because it's got the L of metabolism. But these are things that are made by the cell at various points, and you can find out by what they're making and where the concentrations of things are going up or going down, a lot about the cell's activity. So these discovery-based methods of science are really useful. So the use that we have put them to in this work that I'm going to talk about today, collaboration with people mostly at BYU, but at other places that I'll mention as well, is about using these whole pictures, the discovery-based enumeration of the players in a given biological system to find molecular symptoms of disease. The molecular symptom idea, whoops, wrong button, is from the idea that the way that we diagnose disease is symptoms, but not all symptoms are equally useful. These symptoms seem particularly useless. Please don't waste your time or lose the flow of things by reading that whole cartoon. If you want, I'll email it to you later. It's about how the Simpsons are symptoms of society's problems. But anyway, it was just what comes up on Google image search of symptoms. That's something that's interesting to show. The symptoms that you're used to hearing about when we're talking about disease are called clinical symptoms. Symptoms that a clinical specialist like a nurse or a doctor would be able to observe and be able to use as part of their diagnosis and prognosis for your pathological condition. These clinical symptoms, though, have some severe limitations, one of which is that too many of them are of the too little, too late variety. For example, the subtle signs and symptoms of ovarian cancer have led to it being called the whispering disease. A lot of women that have ovarian cancer don't get diagnosed until they're stage 3 or stage 4, which means that the disease has progressed to a point that the prognosis is unfortunately quite grim. If you could catch the disease at earlier stages, you'd have a much better chance of intervening successfully. But there aren't many clinical symptoms in earlier stages of the disease. So those subtle signs and symptoms that clinically present themselves are insufficient. So we want to look for instead molecular symptoms, because molecular symptoms can come much earlier than clinical symptoms, well before the patient's even aware that they have something wrong and need to go to the doctor or physician's assistant or whoever they're going to see. And also the molecular symptoms, in addition to being earlier, can be much more specific than clinical symptoms. Instead of just complaining, well, I have pelvic pain, could be ovarian cancer, could be endometriosis, could be what you ate last night. Well, what if you're able to identify molecules that are specifically created by cells in response to certain types of damage, or molecules that are even part of the pathology itself that are causative in the pathology? This can mean that you can learn a lot more about specifics of the disease, including the stage of the disease can be elucidated by the molecular symptoms, and even we can learn something about the etiology of the disease so that we can advance our abilities to diagnose and treat. So molecular symptoms are something that we are going to want to look for, but we're going to want to be as uninvasive as possible. You can imagine that if we actually went to and sampled all of the tissues of the body, we'd have a much more robust molecular picture of the body, but the patient would really be hesitant to go through that kind of diagnosis all the time whenever they go to a doctor's visit. So in trying to be as uninvasive as possible, we've decided to focus on a tissue that is more readily obtainable and that has a connection to all of the other tissues, and that is the blood serum. So blood obviously visits all of the organs of the body. Blood is intimately connected with all of the processes of the body. And serum that's produced by allowing blood to clot is produced in a very reproducible way between different clinics and different labs. And this is a really important aspect of trying to get biological samples to look at the molecular level. You really want them to be as reproducible as possible. There are other ways to collect information even from blood other than serum, but they might not be the same at a rural hospital out in some part of Montana as compared to a sophisticated hospital in, say, the metropolis of Orem, Utah. So we've decided to use blood serum, and we refer to it as truth serum because it's going to tell us what's really going on in the body. Okay. The problem with serum, though, is that it's a really weird molecular view of, especially in terms of proteins, a really weird molecular view of the body. Serum is dominated by a very small number of very abundant proteins that I'll call bulk serum proteins. One of them, just one single protein, accounts for 2/3 to almost 3/4 of the total protein in your blood serum, serum albumin. And then others, including immunoglobulins and a few others, account for up to 98% of the total protein mass in your serum. So all of those bulk proteins are found in everybody's serum, and they're not likely to change very much in terms of indicators of different physiological or pathophysiological conditions. So we spent a couple of years in my lab when I was at BYU developing a method to get rid of the bulk proteins and just keep all of the other proteins, which we found that we could see at least 9,000. At first we were seeing about 5,000 or 6,000, but we improved our methods to where we were seeing about 9,000 different protein species in the serum that weren't these bulk proteins, that instead constituted about 2% of the mass of the protein in the blood, but overwhelmingly the numbers of different proteins were still well represented, and we can use these many different proteins that are in lower abundance to get a physiologically relevant and pathophysiologically relevant snapshot of the molecules coursing through the body's circulatory system. So this is the approach that we used for all of these diseases, except one where we actually did use tissue, and I'll explain why that was a different case when I give you the details about it. But we were interested in applying this method to the diagnosis of disease, to looking for proteins whose abundance was changed in a diseased state compared to a healthy state. Either the disease includes an increase in a protein or a decrease in a given protein in its abundance in the serum. And so where do we apply this method? What diseases are we going to look at? The most important decision here was, where can we get good samples? And that meant collaborating with clinicians, or at least with biomedical scientists that had access to samples that we as bioanalytical chemists and myself as a protein chemist didn't have access to. So one of the early applications that I'll talk about first is an application to hypercholesterolemia, and then the majority of the others have been related to women's health, especially the complications of pregnancy, including preterm birth and preeclampsia, and also endometriosis. And the reason for focusing on these diseases is that we had a very important collaboration with the University of Utah obstetrics and gynecology program. I'm actually an adjunct professor in that program, which really fills me with fear, because I worry that if I get stuck in an elevator with a woman who goes into labor, she'll say, "Oh, but you are an adjunct professor in the Department of Obstetrics and Gynecology. Can't you please help me deliver my baby?" And I'll just be in a really bad situation. Because, I had a bad time even when my wife was being delivered by people that knew what they were doing. I was really worthless. And so, anyway, this collaboration has allowed us to get some really excellent specimen banks that have allowed us to do some really super science. And then the last one is a newer collaboration with some people at Washington University and at Brigham Young University that have helped us to apply these methods towards studying Alzheimer's disease. So I'll go through each of these diseases one by one and then talk about the results that we've had up to this point. So the first application I'll talk about is about hypercholesterolemia, having too much cholesterol, which really does show up in the blood. But this project, we didn't use blood. I'll talk about the tissue that we did use on the next slide. The reason for studying hypercholesterolemia is probably pretty obvious to most of you. Cholesterol levels correlate with cardiovascular disease risk, and cardiovascular disease, whoops, wrong button again. Cardiovascular disease is the #1 killer in our society, bigger than all other causes except cancer put together, at least some years. Sometimes in the past couple of decades, cardiovascular disease has been outpaced by cancer, but as we're getting lower and lower cancer, we're getting a bigger and bigger picture, a bigger and bigger slice of the pie taken up by cardiovascular disease. One of the significant risk factors in cardiovascular disease, in fact, the most significant single risk factor, is hypercholesterolemia, where cholesterol, the LDL form of cholesterol, is deposited as plaque in the circulatory system, and that leads to, if it's in the arterioles of the heart, that can lead to heart attack. If it's in the arterioles of the brain, that can lead to stroke. If it's in the lungs, that can lead to pulmonary embolism, all three of which can cause death and are obviously really big problems. So we want to learn as much as we can about hypercholesterolemia. And so the opportunity for this collaboration came about because one of my collaborators at BYU took an airplane, which is always the start of a good collaboration. He happened to sit next to a woman who was also a scientist, a biomedical scientist at the La Jolla Institute for Molecular Medicine. Her name was Carol Benka, and as they flew along, they told each other about the work that they were doing, and it turned out that Dr. Linford, in work that he and I were starting up to look at the various lipids in tissues, we could apply the methods that we were developing to samples that she had from hypercholesterolemic mice. So there were 4 different groups of mice that had a normal diet, mice that had a normal diet but were knocked out for a specific gene, and this is why these experiments were being done in mice. The knockout mice with a high-fat diet that then were reverted to a normal diet, and knockout mice that were allowed to just continue on a high-fat diet. And it was found that mice from these first 3 groups had normal amounts of cholesterol, while mice from the knockout with high-fat diet had very high hypercholesterolemia. And so we analyzed the liver samples from 50 different mice from these 4 different groups. We were blinded as to which group the different mice were in. We observed about 1,100 lipids in each sample, and then our goal was to group the mice according to their lipid profile and see if we can make any sense about what lipids are up or down regulated because of having high cholesterol. So using lipids as markers. Now, before I tell you the results of any of the experiments, I'm going to tell you about what the other problems are that we worked on. The complications of pregnancy. We decided to focus on, this was in collaboration with our clinical collaborators at the University of Utah, the 2 biggest problems that are complications of pregnancy. The one that kills the most babies which is preterm birth. So preterm birth kills more babies than any other contributor to neonatal death. And so we obviously felt like that was a very important problem to get at. And it's very difficult to tell if a woman's going to have preterm birth until right before she actually goes into premature labor. So this study was made possible because the clinicians at the University of Utah are part of a large network called the Maternal and Fetal Medicine Unit Network, or MFMUN, which has nothing to do with the United Nations. That is a part of the National Institutes of Health. The group collected samples from almost 3,000 women at 14 centers across the United States, including the University of Utah. Blood was drawn at 24 weeks of pregnancy and 28 weeks of pregnancy, and then the patients were followed to see when they delivered. Premature birth, preterm birth, is counted as birth before the 37th week of pregnancy, and we had blood samples from up to a couple of months before that. The reason for these collection of these 3,000 samples was that they already had some hypotheses about what things might correlate, what blood, well, it was blood serum, components might correlate to preterm birth, and they had a list of about 30 compounds that they were going to evaluate. So after they'd collected all of the samples, they evaluated all of the specimens, and they found that none of their 30 candidates statistically changed in abundance with preterm birth. In other words, they did a hypothesis-driven science and found that their hypothesis was wrong. Wow, that was a lot of money and a lot of difficulty to go through to find out that you didn't know anything. So we were able to take many of these specimens. We took 140 at first, 40 case and 40 control. That is 40 women who went on to deliver prematurely and 40 women who went on to deliver after 37 weeks at each of the first visit at 24 weeks and the third visit at 28 weeks. So then we analyzed all of the proteins in those samples, and we came up with a list of candidate biomarkers, which we then studied in a validation study with another 100 patients' samples. And these we were blinded to. We didn't know before starting the study which samples were from women that went on to have preterm birth and which were from women with normal pregnancies. We observed, like I said earlier, more than 9,000 proteins and peptides per sample, and we statistically correlated changes in abundance with the outcomes of the pregnancy. We also decided to study the #1 killer of women in pregnancy, which is a disease called preeclampsia. Preeclampsia kills more than a third of the women that die perinatally, and it is very poorly understood in terms of etiology, what causes the disease, and you usually don't find out about it until it gets to the point that it is difficult to treat. One of the real problems with preeclampsia is that the first clinical symptoms are really general symptoms like high blood pressure and swelling, and I'm sure that you've all known women who, while expecting, experience some high blood pressure and some swelling. Those are pretty normal symptoms of pregnancy, and then when those symptoms are accompanied by proteinuria, protein in the urine, and some other clinical symptoms, then a diagnosis of preeclampsia can be made. In fact, by the time you get to proteinuria, you're usually in fairly severe preeclampsia, but it's difficult to know where to draw the line in terms of the clinical symptoms. So we looked for molecular symptoms, and this was a part of another study that the University of Utah was a part of, called the FASTER study, first and second trimester evaluation of risk for preeclampsia. Samples were collected from women that were pregnant at 12 to 14 weeks of pregnancy, so this is much earlier than the samples in the preterm labor study, and then the second visit at 16 to 18 weeks. And this was a much bigger study, over 10,000 specimens taken from women, from thousands of women at a number of different centers, including the University of Utah. We began these studies by looking at 100 specimens from women that went on to have preeclampsia, and then from 100 controls that had uncomplicated pregnancy. Again, we observed almost 10,000 proteins and peptides, and we correlated the changes in abundance of some of these proteins and peptides with their pathophysiological outcome. The last gynecological disorder that we looked at, which is a much more recent set of experiments that really we're still in the throes of bringing to conclusion, is relevant to a disease called endometriosis. Endometriosis, which is very unfortunately common, 1 in 20 to as much as 1 in 10 women experience it. In fact, it's very difficult to know exactly how widespread this disease is, because a lot of women have it without knowing that they have it. What the disease is characterized by is the growth of endometrial tissue outside of the uterus. The endometrium is the lining of the uterus, and this is the tissue that's sloughed during menstruation. And some of these cells are able to find their way outside of the uterus and begin to grow in other parts of the body. It's a very common cause of pelvic pain. It's a very significant cause of infertility in women. And as I said, a lot of women that have the disease don't even know it because they attribute these general symptoms to other things. The only way that you can really determine that this is what a woman is suffering from is to be quite invasive and do at least exploratory surgery, in which case the the growths are usually removed as well. The chairman of the Department of Obstetrics and Gynecology at the University of Utah, Matt Peterson, is the principal investigator in this study, and we've so far used samples only from the University of Utah. 24 samples from women, a dozen cases and a dozen controls, where we got not only their serum but also their urine, and we're looking at we haven't yet established a protocol for ways of looking at the urine. But with the serum samples, again, we observed almost 10,000 proteins and peptides from each specimen, and then correlated changes in the abundance of any of them with the endometriosis status. And the last disease that I'll refer to is a disease that is growing in its significance in our society, and that's Alzheimer's disease. As our society lives longer and longer, diseases of aging are obviously taking their toll on us. But some of these age-related diseases, if we correct death rates for age, then we actually see that we're making headway against some of these diseases. Cancer, heart disease, including stroke, even diabetes, we're actually having fewer deaths just in the last decade or so as a percentage when corrected for age. But Alzheimer's disease is on the increase, and so this is becoming more and more of a significant problem. And it's a very difficult problem to diagnose. The symptoms are behavioral, and so they're hard to pin down, and they often don't get flagged by the patient or the patient's family or by clinical attendees until the disease is at a fairly progressed stage. And it would be very useful to be able to get molecular symptoms for this disease. So, in collaboration with Dr. John Coway at BYU, who I understand was a presenter yesterday, I had classes all day yesterday and wasn't able to attend, but he did another collaboration with UVU going to Samoa, but he was a graduate student at Washington University in St. Louis, and has access to their clinical programs there, and he does basic biology of Alzheimer's disease, and he secured for us about 50 cases, the serum specimens from about 50 cases of Alzheimer's patients, and 50 age and gender and ethnicity matched controls that were all collected at the Washington University Medical Center in St. Louis. And we observed almost 10,000 peptides and proteins, and then correlated the changes in abundance of these proteins and peptides, first of all, with whether or not they were the cases or the controls. when we found that we were actually easily able to identify biomarkers that could tell us whether a given sample is from an Alzheimer's patient or a normal patient, we decided to look at the medical records of the patients and correlate with the disease status, whether it was advanced Alzheimer's or earlier stage Alzheimer's disease, and also to look at gender, because Alzheimer's disease progression, incidence, frequency, and progression differ really significantly between men and women. So, now that I've set the stage, I'm going to tell you about how things turned out. First of all, in the hypercholesterolemic mice study that we did with the La Jolla Institute for Molecular Medicine, we were blinded from the beginning as to which mice the samples came from. These are liver samples, and we looked at lipids, and we found that we could easily separate them into two groups, the majority in this group and a minority in this group over here, based on, I won't go into the analytical methods, it's called principal component analysis, but we found that this group were the mice with the extraordinarily high levels of cholesterol, and the other group were the mice with normal levels of cholesterol. So we easily could see, and we could see cholesterol, but we excluded it as a marker because that wasn't the lipid we were looking for. We can go about our business, move along. You have to be a Star Wars fan. Okay, got lost there. What we found that were markers that did significantly correlate, both being increased, some of them, and being decreased, were molecules like the one shown on the slide here that are called lecithins or phosphatidylcholines. And this actually makes a lot of sense. These are membrane components, and cholesterol's most important function in the body, and where we actually find the majority of cholesterol in the body, is in membranes. It regulates the fluidity and rigidity of membranes, and what the body is obviously doing in response to changes in cholesterol is changing other membrane lipids abundances to change the membrane rigidity and fluidity that now has been thrown off by the different cholesterol. So we could use these lipids, which do actually appear in blood, as possible biomarkers of hypercholesterolemia, or more importantly, to understand something about the etiology of hypercholesterolemic disease. I won't talk about the details there. So now on to the complications of pregnancy. First, preterm birth biomarkers. Here is an example of a biomarker where we have 8 mass spectra. I don't know if I even mentioned that the way that we observed those thousands of proteins and peptides was on a mass spectrometer. So we're doing capillary liquid chromatography, electrospray ionization mass spectrometry, which enables us to observe individual molecules by deducing their molecular mass. And so this is a compilation of 16 different mass spectra. The 8 mass spectra that are in red, that in this region of the spectrum have very low signal, are from preterm birth, women that went on to have preterm births. The 8 in blue that have a, this is one molecular species that has different different isotope forms. That set of spectra are from women that had normal, uncomplicated pregnancies and didn't deliver preterm. So this molecular species that causes this signal is dramatically increased in women that have normal pregnancies and lost in women that have preterm birth. We found about a dozen such candidates. 5 of them that were very good statistically were from one protein, 3 from a single splice variant of that protein, and 2 others from another splice variant. And using these different markers, in fact using only the 5, we got over 90% predictive value in terms of being given a blind sample and being able to classify it as a woman that had preterm birth or not. And remember that these samples are being taken several weeks to months before the woman actually delivers. This technology was patented, and then the patents licensed to a startup company that is located at Research Park in Salt Lake called Seroprognostics. This company has recently made a big decision. They developed these markers as a test that they have trademarked as pre-TRM, for preterm, right? They've decided that instead of putting it on the market right now, they have enrolled 5,500 women in a prospective study using those markers so that they can get a level of FDA approval that will allow them to be as clinically relevant as any diagnostic test can be, where hospitals and doctors can prescribe this for their patients. And so that prospective study is underway right now. With respect to the other complication of pregnancy, preeclampsia, here are a number of markers. Again, the red are in this case, the red are the normotensive women, the women that didn't have preeclampsia, and the blue are the preeclamptic women. And we can see markers that are increased in preeclampsia and markers that are increased in normal uncomplicated pregnancies. We found more preeclamptic markers than preterm birth markers, and we can, by different statistical analysis, give either 100% specificity and 83% sensitivity, or we can get as high as almost 90% sensitivity with lowering our specificity a little bit. Specificity means the frequency of false positives, and sensitivity means the frequency of false negatives. And we find that combinations of the markers are the best. We licensed this technology to Seroprognostics as well, and they're in the process of developing commercially available diagnostic markers. One of the markers that we identified here is a fibrinopeptide. That means that it's involved in coagulation, and there is a known coagulatory problem that relates to preeclampsia called disseminated intravascular coagulopathy. And so the relevance of this marker biologically seems to make a lot of sense. Endometriosis is a newer project, but thus far we have identified 20 markers with statistical p-values of 95% confidence or higher, and almost double that with a looser statistical confidence, but we can increase the confidence by using those, what you might term lousy markers, in combination, where our statistical significance goes up. Interestingly, one of the markers is a gene product that the mRNA of which has already been implicated in endometrial tissue. So there's some biological sense here. In Alzheimer's disease, we've also identified several different markers. So we have the red, which is, I think, the diseased here, dominating over the blue, or the blue dominating over the red. This shows no statistical significance, so this is a control protein that's the same in people with or without Alzheimer's. And we found about 36 different statistically significant markers. When we found these markers and they jumped out so clearly at us between Alzheimer's versus regular patients, we decided, hey, can we say anything about the degree of the disease? And so we re-evaluated and found that we have several markers, about a dozen, that change between early and late stage disease, so that we might be able to not only diagnose disease, but even say something about the stage. And very interestingly and importantly, we have markers that are gender specific, as well as some that aren't. And this might help us to understand more about the etiology of the disease and why, in fact, there is such a different frequency of disease with the two genders. So I hope you can see the promise of these various, applying these methods to these various diseases. If we have the right clinical samples, clinical specimens, we should be able to apply this method of looking for biomarkers to virtually any disease that has contact with the blood, which is virtually virtually any disease. In closing, I'd like to give appropriate credit and gratitude. I'd like to thank the people that gave us the money for these studies, including Utah Valley College of Science and Health that has given money, especially for the students that have traveled to various meetings to present these results over the years. Collaborators at the university, at Brigham Young University, where I started these studies before I came to UVU, Dr. Stephen Graves, who's a clinical biochemist over at BYU, Dr. Matt Linford, an analytical chemist at BYU, John Cowie, a biologist at BYU, and Evan Johnson, a biostatistician over there. At other universities, Dr. Sean Esplin at the University of Utah, one of the leading high-risk pregnancy OBs in the nation, and really the initiator of our application of these methods to pregnancy complications. Dr. Matt Peterson, the current chair of the Department of Obstetrics and Gynecology at the University of Utah, and Dr. Carol Banka at the La Jolla Institute for Molecular Medicine. BYU graduate students that worked on this, on these various projects, and BYU undergrads, too numerous to mention. University of Utah Valley University undergrads that have worked on this project are listed here, and it's been a joy to work with them. They've been very kind over at BYU to let me take UVU students over there, even though a few of us have had beards and so forth, and they've been very helpful. My hope is that we can get some proteomics work going here at UVU, and Dr. Heaton and I are trying to start those kinds of studies over here. Thank you for your attention, and I'd be glad to take any questions.
We have time for about one or two questions.
So I'm interested in how does genomics and proteomics talk to each other? You know, because genomics with disease are looking for single nucleotide polymorphisms, right?
Right.
And those that are affected have that mutation, and those that don't. Because your methods don't pick up on those proteins that have a single change, or they do or don't?
What we find is proteins that are the same protein in a person that's affected or unaffected. So the gene for it is the same.
Right.
But they're expressed at different levels in a person that has
So that's what I'm
disease
wondering,
versus
is how does
not.
the mutation...
So there may be a mutation in the regulatory regions of the gene, or more likely, there are differences in the epigenetic circumstances that control that gene's regulation.
Right.
Also, almost all of the markers that we've identified are fragments of proteins. They're not full-length proteins, and so there's post-translational processing that's an important part. The only slide I skipped is about the fact that this one protein that appears in as 5 different markers, 5 different parts of it, from 2 different splice variants of this protein in preeclampsia. It also appears in preterm birth and is one of the markers in Alzheimer's disease. It's a protein that people have known for decades is involved in inflammation. What we didn't know before these studies is that it's processed differently in different inflammatory diseases, different diseases that have inflammatory response as part of the disease. So there are details of biochemistry that we can only see by looking at these kinds of products.
So is it fair to say then that the genomics is looking more at the genes that are controlling the final product, and you're actually capturing what the picture looks like in the final product?
We're looking at
where
the parties instead of just looking at the invitations and guest lists.
Okay.
That's the idea of proteomics.
You don't have to answer this. It seems to me, though, that, and I love the research, and I'd love to ask questions about it. It seems like to me, though, in education, we're missing out on some of the skills that are required for this kind of research. In other words, our classes don't focus as much on the statistical analysis as they need to for this type of research, and even some of the biochemistry is weak for understanding.
Well, we certainly do,
proteomics.
as education has for centuries, focus on hypothesis-driven directions. That's because we have the conceptual orientation in academia. right? So if we have ideas, it's harder to teach. It seems more meaningless, actually, to teach, to just go out in nature and enumerate what there is.
I sort of understand that. I don't know that I agree with that thought completely, in that I think there are ways that we can, I mean, really what this leads to is a working model of how something functions.
The discovery-based science will always help us to make better hypothesis-driven approaches.
So it seems to me like we ought to, and all but I don't know these guys, it seems to me like we ought to make a more intentional focus in our lab work and even in some of our classwork.
On
These types
discovery-based,
of methods and applied
I would agree with you that that's a fruitful direction to go. Thank you all for your attention.
We are, in fact, bringing up a statistics degree next year
Oh,
on
excellent.
campus, and we are hiring a biostatistician. So
So the real thing though is we need to integrate.
there's no question. Yeah. Thank you, everyone, for coming.